Good morning, everybody. Hopefully everybody had a restful night. Good morning also to all of our virtual participants. Looking forward to another exciting day of presentations and discussion. This morning is going to focus on image informatics training and education, and then after that we will have core facility management training and education. And then after that we will have a session after lunch on data management. And our final session this afternoon will be around impact, which will highlight some of the members from our community who are really exemplifying the work that we're all trying to do. Then at the end of that, we will have a poster session, which unfortunately will not be able to have our virtual audience participate in, but that is the program for today. And we will aim to try and keep things on track so that you'll have a nice break in the middle of the day to just regroup. So with that, I will pass things on to Claire Brown, who is co-chair of BINA, to moderate the first session.
So thank you, Nikki, and welcome everyone this morning. So our format for this session is the same as we had last evening with Phil Hochberger and the panel on education. And so we'll have 3 presentations, and then we'll have a good 30 minutes to have Q&A from the audience. So get your questions going as you see the presentations. So our first presentation is from Beth Cimini from the Broad Institute.
Good morning, everybody. Everybody have a good night's sleep? Everybody awake? Hey. All right. Good morning. I'm going to tell you guys, so I'm Beth Cimini. For those of you who don't know me, I'm the Associate Director for Bioimage Analysis at the Broad Institute in Cambridge, Mass. In the US. And I'm also a member of BINA's Image Informatics Working Group. And I'm going to tell you just a little bit about some bioimage analysis efforts that we have in the US, but because there aren't all that many that are actually specifically in the US, some of the global initiatives that we're trying to connect the US and other regions to so that we can really make bioimage analysis a global thing that we all can contribute to. And I want to start with this slide because I think I've said this to a bunch of you who are here in person already this week. Image analysis is at a little bit of a disadvantage compared to a lot of other quantitative sciences in the fact that nobody ever analyzed a single-cell RNA-seq experiment without a computer. But we have about 400 years of history of looking at images down a microscope and, creating beautiful images like this that I could not create, but judging what happens in microscopy by what our eyes can do. And we have maybe 40 years of experience of sticking these things through computers. And so there's a little bit of a consideration that image analysis is a bolt-on or an add-on, because historically it is, but that doesn't mean it necessarily should be going forward. And this paper is about 10 years old, but I'm not sure it would change that much now. So in papers that called themselves high content screening, and high content screening, you're typically talking about sort of large scale images and or getting lots of information from images. Well, more than 75% of papers at that time only reported 1 or 2 cellular features. So it might have been something like the cell size or the cell intensity, but only 1 or 2. Now, if you have a 4 megapixel camera, which is pretty standard these days, that's 4 million pixels every time you snap that camera. And you might do multiple channels on multiple Z-planes and multiple time points. So you have conservatively, sometimes in the tens or hundreds of millions of pixels of quantitative information, and you're reporting 1. So we want to work on changing that because we think there's a lot of information that's just simply being left on the table that we could do more with if we were able to extract it. And certainly within the pharma space, I apologize for those of you who saw some of these slides already. Big data is really what people are hoping is going to start coming out. And so this is from the SLAS, which tends to be a pretty industry-heavy, high-content imaging special interest group from a couple of years ago. And when we ask people what their hopes are for the future of this field, a lot of it is around ML and big data and, just lots and lots and lots of data that we can then use in these new quantitative ways. And again, these are mostly wet lab microscopists who are attending this thing. And when we asked them what they wanted to hire for, they said Python skills. So there is this big gap in the community that industry definitely is noticing, but academia is still a bit behind in terms of we are able to now generate a ton of data. There's stuff that's there. We want to mine it, and we need more bioimage analysis in order to be able to do the kinds of science we want to do going forward. And so hopefully academia is going to start catching up to where industry already is. The problem being, again, that if you are a scientist who spends most of your time in the lab generating data, when we ask people in a survey how comfortable they are in sort of generating, developing new computational skills, their amount of time doing imaging versus image analysis is left to right, and their computational comfort is up and down. And it's maybe not surprising that the more time you spend on a computer doing image analysis, the more comfortable you are with computing skills. But it means that we have the people who are over here who are generating data and who are actually doing the experiments and making all of the tiny decisions that, you know, that determine whether or not an experiment is actually analyzable or not, who don't know what the eventual computational workflow is or don't understand how it's going to be used later often. You know, certainly there are some excellent computational people in the wet lab, but you're more likely to not have a good understanding of how that data is going to be used. And so you might make decisions that are going to harm the usefulness of this data later. And we did a similar poll for those of you who were at the EOE yesterday. For those of you who were, thank you. We loved having you. But from previous BINA EOEs we've done, people say who are in facilities, when we ask them what is blocking them from providing more image analysis education to their users, you know, we are so grateful to all of the core facility staff who train so many people in microscopy and imaging and sample prep and all of those amazing things. But when we ask, do you wish you could provide more analysis education? Everybody says yes. And this is why they say that they can't do it at the moment. And it tends to be experience, and it tends to be time for them to do it themselves or time for them to generate that experience in order to be able to actually do this. And so we need to be able to change how things are done or what things are available so that we can remove this block. And especially, we really see in image analysis core facilities as a major way of distribution of image analysis best practices and tools, because you are the folks that people trust. They come to you, you teach them how to make their sample, you teach them how to image their sample and make these images. They will trust you also if you tell them this is how you analyze them. You know, those weird computational folks off in the corner that they've never met before, they're much less likely to come to us and ask us questions. You guys, if you can be the disseminators and the distributors of this information, it's going to go a lot further and sort of penetrate a lot deeper. So what do we actually need to do this? So the good thing is that, so I stole this from Robert Haas's Twitter account. There's starting to be a really global network of certainly bioimaging groups forming. And there's a few bioimage analysis groups in there too. So I've marked these guys with stars. These are groups that are specifically bioimage analysis groups. There are 4. And I think it'd be an interesting question for the discussion later. Should we be actually thinking about, do we want more bioimage analysis societies, or do we, in the same model that BINA has, want image informatics to be part of just a larger bioimaging group? I think there's pros and cons in either direction. And so I think we really want to think about how we want to do that going forward. But now I'm talking to the image analysts and image analysis tool creators in the room. What we need to do to help you is to make better tutorials. And especially when we ask people who say they spend most of their time imaging, what they want tool creators to do for them, they want us to just make them some videos. They really, really, really just would like some videos, please. And they would like things in general to be easy and friendly. I think it's interesting to say that, or just to show sort of the opposite thing as well, though, which is when you ask the opposite group, when you ask the tool creators what they want users to do, you know, you get some words like learn, but a lot of it is just words like report and share and feedback. If we don't know that a tool isn't working for the community, we can't make it better. And so encouraging people to give feedback, to go on the image.sc forum and just sort of tell us what's not working for them. People care really passionately about making these tools better, and we want to make them better. And so we would love more feedback. In general, if we look at sort of the mechanisms and kinds of ways that we could convey this content, people tend to prefer sort of written tutorials and then video tutorials and things like that. But I just said that people want videos, and the reason that that ends up coming out is if you look at stratifying all of this data, and all of this data, I should say, is from a paper that's about to be published in Journal of Microscopy, but all of the survey data is available online, and you can check it out yourself and take a look and see what people who have a certain profile want from you. Written tutorials, everybody's like, yeah, no matter what their level of self-reported computational skill is, they're totally fine with, you know, a written tutorial. But video tutorials, people who describe themselves as computationally high skill don't really like them, but that's what people who describe themselves as computationally low skill really want the most. And so we need to do a better job of sort of not making what we ourselves, the developers, want, but what the actual people in our communities want. They would also especially like to come to office hours, which again shows the same split between high skill and low skill. And so there's starting to now at least be some guidelines. There's a lot of people working really hard on this problem and who understand where the gaps are and are trying to fill it. I forgot to change that this is no longer on archive. This is in Nature Methods now. So QuARP Working Group 12, which is about image analysis, just recently put out some checklists for how to report imaging as well as image analysis in publications. And we're hoping that journals and funders sort of get behind these checklists or help us make them better and start, you know, making them actually enforceable. But if people are coming to you and saying, I don't know how to even report the analysis that I did, we're hoping that this provides a guideline where you can give people and say, you need to at least do this much. And we recommend, if you want people to follow the work that you did, to actually sort of go to these further recommended and ideal steps. And so to sort of wrap up with talking about the thing that I said I was going to talk about, which is actually who are the groups that are doing this and how and why, I really want to commend Bioimaging North America's Image Informatics Working Group, because it is the biggest working group of image informatics people that I am aware of. And it's not particularly North American. I would say it's only about half North American. So if you're not North American and you're really interested in image informatics, it is really one of the biggest and best and friendliest places to start doing this. And of course, BINA works closely with Eurobioimaging, Global Bioimaging, and we're starting to now cross-pollinate with Africa Bioimaging and a bunch of the other ex-bioimaging groups as well. And so we're happy to work with those groups and sort of expand our reach, because again, we know that this is a global problem and we know that people want to do this everywhere. I want to especially highlight one particular thing that's being sort of run by BINA and COBA and New Bias, New Name Pending, which is the Halfway to I2K event that's going to be happening in October. So we're going to have a virtual online tutorial-a-thon. We're asking developers and people who are really good at using tools to submit tutorials and tell us, and just volunteer to teach whatever they want to teach at whatever time they want to teach it, and just go out there and say, I'm going to give you this information in whatever language they want to teach it. At some point in this Zoom platform between October 17th and 19th. And then we're just going to open it up for free for anyone who wants to come. And this model, as part of the full I2K meeting in 2022, was really successful, and we hope it will be in the next full I2K meeting in 2024. But since there's 2 years between meetings, at Halfway to I2K, we still want to make sure that these tutorials are available. So if you are a person who's really good at an image analysis tool, please sign up to teach. Sign-ups are open for at least a few more days. And if you're a person who just wants to learn more about an image analysis tool, just come sign up and take the tutorial of the tool that you're interested in. I mentioned COBA really quickly, and so I just want to sort of give a little bit of a shout out to that, which is a group that's being run by the Cell Profiler and ImageJ teams. We work on making new technology and making new community engagement materials, working with biologists who have interesting and driving biological projects. And so we're really excited to find people who have really hard biology that we can make new tools for. So if that sounds cool to you, come talk to me, because we would really love to help make workflows that makes your analysis possible if it's not currently. And sort of now moving off into some other continents, AI for Life is a Horizon Europe funded group. They're having open calls for helping researchers where, again, you can say, here are my images, here's my analysis problem, help. They're doing hackathons for tool creators. So keep an eye out for those things. Sign up for their newsletter. They're the ones supporting the Bioimage Model Zoo with models of deep learning for lots of different things. So please go sign up for their newsletter and check them out because they're doing amazing things. New name pending of New Bias. Finding a name for something is really the hardest part. Has been doing a great job now for several years, and we're really excited that we're refunded and have a new steering committee, and we're about to be off and running. They do these amazing multi-day training schools. There's an annual image analysis conference, but they also make things online for benchmarking called Bioflows. And the Bioimage Informatics Index, which has hundreds of different image analysis tools and workflows where you can look and see what might work for your data. So please go check out those resources. If you want to get better at learning how to teach these sorts of things and give trainings, especially around computational topics to biologists who aren't necessarily super receptive to computational topics, I've found life science trainers, which tends to be a lot of folks from sequencing and bioinformatics, but they are also solving these same problems of teaching computational topics to non-computational users. And so I'm trying to get a few more image analysis people to come hang out with me there. So it's a really great resource for figuring out how we teach computation better. And if you forget everything else that I said, forum.image.sc is the online scientific community forum where we have more than 60 open source image analysis tools that do all of their support in one central location. So if you have a problem with a particular tool, you can come tell us that. If you just have images and a question and you're like, I don't know what to do, please help me, we will do that too. That's exactly what the forum is designed for, and we want more beginners on there. There are no bad questions, there are no stupid questions. There's 30,000 answered questions on there if you want to check it out and see if your question's already been answered. But if not, we would love your question, no matter whether it's beginner level or incredibly developer nitty-gritty level. And so to just close out, we really see that one of the biggest global problems is bringing image analysis and data interpretation into this cycle of, it's not that sample preparation and image acquisition go back and forth, and then you bolt this half of the circle on at the end, but that we start to see imaging as a fully quantitative science and sort of bring this into the cycle and let it feed back all the way through experimental design. And so how do we make people see it this way? How do we keep this from not being an afterthought? And how do we tell people who don't know that they don't know this, that they don't know it, and help them learn? And so these are the things that I think this room is going to be great for solving. And so with that, thank you to the people who give us money and the awesome people in my lab who make cool things that I got invited to talk about. Thanks.
Okay. Beth, for that fantastic talk and for pointing us to so many great resources in the image analysis area. So our next presenter is Paúl Hernández Herrera, who is with us from the Autonomous University of San Luis Potosí here in Mexico. Oh,
Here.
oh, you're right here.
Thanks, everyone. Good morning, everyone. Today, I am going to talk about imaging informatics training resources in Mexico. So I want to say that this is not my work. There is a team behind me. So I am going to talk what I have done the last 2 years, that I have been working as a CCI imaging scientist. This work is thanks to the CCI award. So this award allowed me to join to the Mexican National Laboratory for Advanced Microscopy. Here, I was working by myself in a group. Now, when I joined to the Mexican National Laboratory for Advanced Microscopy, I have the opportunity to join an extensive network of biologists, and I really see all the resources that are needed, especially training for biologists. what I have been working is just to try to create tools to make AI and artificial intelligence more accessible for biologists. So I have been working on online courses and tutorials, in-person workshops and training, custom AI routines tailored for biologists, working on user-friendly tools and interfaces. And what I always say is that biologists should use all those tools that we have available. So they are really, really nice tools that you can use. You just can go, just upload your image, and you are going to have something that it is automated, and it is going to get very, very good results for image analysis. Beth already gave some really, really nice tools that the community is working. So I really encourage to do that. So something important for me, express myself in Spanish is difficult. It is my native language. Expressing myself in English is even more difficult. So all the courses are in Spanish. So this is something that I say, it is better for me to explain all these courses in Spanish. People, it's going to feel more comfortable with the language. So all these courses all these topics that I am going to explain are in Spanish. So it is not for English, it's only for Spanish people. As I say, I have been working on online courses. So what usually happens is that I have a big community. Chris Good is also, he makes a lot of networks, and he say, okay, I have a person or a group that wants training in some topic, and I say, okay, let's design something that they are really interested. So for example, we have the opportunity to give a course on introduction to image analysis. We don't talk about special things, mathematics, all those topics related to algorithms. We just try to give something that is really user-friendly. If people doesn't know how to use Fiji, we are going to give a course related to Fiji and step by step let the people know how to use these tools. It is not for advanced people, it's for people who wants to introduce or want to get a feeling on how to use all these tools. So for example, this course that we gave, Introduction to Image Analysis, we give topics related to image analysis, digital images. I work with some other friends, and he gave some other topics, image filtering, fundamental of fluorescence, and image processing. And that was only the basis of image of Fiji, just to get the feeling how to use all these tools. So Adam, he has also helped me a lot with these Mexican bioimaging workshops. They have a topic related to also image analysis. Every year they are organizing courses on image analysis, just the basics. And we are planning to organize also a workshop on artificial intelligence for image analysis. Note again, all these courses are in Spanish. I have also worked on online, as I say, Jupyter notebooks are very, very easy to use. You don't need to implement anything. You just need to use the Jupyter. You implement everything. The developer implement everything, and the user only needs to go and say, okay, I'm going to give click, and they are going to obtain the result. So I am working with deep learning for 2D segmentation, 2D classification, 3D segmentation, and we just create these notebooks, and they are really, really easy to use. You don't need to do anything, just use it. In-person workshops. So we have the opportunity also to attend a course on the National Laboratory for Advanced Microscopy. The Ripicot is another CCI imaging scientist. He gave a course on multiplex images, and we were able to reproduce this course for another group that was interested on learning this topic. So we make the connection. We try to develop this course. We talk about supervised machine learning. As I say, I don't go directly to the theory. I just give a brief introduction, and they point to tools that they can use. For example, for segmentation, they can use Elastic. For training deep learning models, they can use zero-cost deep learning for microscope. Application of supervised learning, they can use Fiji. And all those topics are tools that people can use, and all those topics, we can also reproduce these courses. As I say, we also have courses related to image analysis on the Institute of Biotechnology. Again, we try to bring all those topics, deep learning, supervised machine learning, just to automatize every image analysis. We have this course on image analysis. I give the topics about supervised machine learning, always the basics. Again, I use webcam and Elastic just as interactive tools to let the people to use all those tools, and they only need to use some clicks and they are going to obtain the results. Again, Jupyter notebooks, for me, Jupyter notebooks are really, really useful. They allow me to code everything, and people just go, biologists or people who are not experienced with this, with programming, they just need to go click, 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 and they obtain the results. Again, for segmentation, we have tools, start this. This is one topic really important for me. I try to teach the biologists to say, you need to only upload your image and you are going to obtain your results, nice cell segmentation with start this, and then you can do all your analysis. It is not always the tools, but they are also tools for manual annotation. People need to know all those tools because they are over there. People need to know all these topics, and also annotation is really important if you want to train your own algorithms. Some collaborations also. Collaborations for me are really, really important. So sometimes they are projects that are not easy to solve. You need to develop something new. You need to apply something different. So how work collaboration with us? Well, for example, some biologists come, identify the problem, and they say, okay, I know that I have this problem. I have tried to use some different tools, and I am not able to solve my problem. So they engage. So Dr. Chris makes a lot of work just letting know that we have this expertise on artificial intelligence, on image analysis. So they approach the National Laboratory for Advanced Microscopy to search for solutions. So I am there, Adan is there. So we have some good people who can help. So they engage, we have meetings. All this assistance is for free. They don't need to pay anything. So they just come and just make collaborations. So we try to make these collaborations, just try to solve the problem. If we have, if we achieve somehow a good solution for the problem, so the reward for us is the publications, or just to create something, a new tool, and that's the reward for us. So that's how work collaborations. As I say, I have worked on several things to do all this possible. For example, we developed this multiplex image analysis. For 2D cytometry, we have been working on segmentation, classification. We have also problems with 2D cell segmentation. Sometimes we use cell post, sometimes we use studies, some different projects, some different models. We have been working on 3D segmentation also. These are really, really difficult problems. For example, for Arabidopsis roots, segmentation of 3D cells is a really difficult problem. And we have used the tools that are available on the internet, but they are not able to solve the problem. So we need to develop something else. For sparse segmentation, this is some topic that there are only few models over there. And we need to do all the training. So also work for fluorescent signal tracking, and all these are collaborations. Clustering, we are working on a very interesting project related to clustering. We cannot solve it directly with the tools that we have, so we need to develop something else. And what is important for me, as I say, I have a large network of biologists. I think that my network is really, really big. So, but now I need to expand my network of AI experts. So on the National Laboratory, I was working by myself with Adan, and now Jonathan, he is also joining to the lab. So I think that is a good opportunity for me. I was really happy to have Jonathan, because my AI network was growing, so it was really, really good, but I have to move to the Autonomous University of San Luis Potosí. So again, I am going to be by myself over there, so I need to expand my network. So it is really important. one person cannot solve all these problems. We need to have a big network of AI experts. As I say, I try to use to develop user-friendly tools. I have developed deep learning for segmentation, Jupyter notebooks for 2D, 3D, and 2D images. These are really, really nice to use because you only need to give clip. You don't need to do anything else. You don't need to know anything about deep learning. You just need to your input image and your output, and that's all that you need to do. It is not only related to Jupyter notebooks. I have also developed custom tools for tracking. This was probably 3-4 years project that I worked a lot. It was really, really difficult. And some other projects related to expert segmentation. As I say, using user-friendly tools, these Jupyter notebooks, you only need to give the input and the output. Where are the images for the input and the output, and the algorithm is going to do everything. They are really, really easy to use. You don't need to do anything, and just give input and output, and that's enough. So why I am developing this? Because when I was working with Zero Cost, it is a really nice tool. You can use it, but you need to upload your data. And sometimes I was giving a workshop, and for some reason, this Jupyter notebook crashed. And I say, okay, now what I am going to do? What can I do? I have to stop. I have to stop at that point. And I say, okay, I want to have control of everything of the Jupyter notebooks. I want to make it them work. So this is going to be my basis for the workshops that I plan to do. As I say, I encourage biologists to explore AI, including deep learning. So just try to use deep learning. It's really easy to use. There are different tools. There are many things that you can do with deep learning. Just try to do it. You can feel to contact me. So this is my mail. If you want to try some, if you need some tools or you want to require one workshop, just feel free to contact me, and we can design something for you. Okay, I think that's all, and thank you.
So thank you very much, Paul, for telling us about all these tremendous resources, and especially having them available to the Latin American community in Spanish. It's really great efforts. So we've heard from the United States and we've heard from Mexico. We're going to round off the session hearing from Canada. So our next speaker is Judith Lacoste, who has an independent company, Cellavie. Welcome.
Thank you, Claire. Thank you, everybody, for being here this morning. Thank you, LABI and BINA organizer, for this fantastic meeting, and thank you for allowing me to present our work that we've been doing under the umbrella of Canada Bioimaging. All right, so we are interested in fostering automated, open, reproducible, and fair science in line with the bioimaging communities and the standard. In 2019, the ISO standard organization published the confocal ISO norm 21073. And as a spin-off from this publication, there was an organization was created, which is called QUAREP-LIMI. QUAREP stands for Quality Assessment and Reproducibility for Light Microscopy Instruments and Images. So QUAREP-LIMI is trying to foster the implementation, the adoption of the confocal ISO norm internationally. Now, there are a lot of us involved in the project I'm talking about today that are members, very active members of QUAREP-LIMI, but also active member of Bioimaging North America. And a lot of these people were also co-author on the Nature method issue, focusing on reporting and reproducibility for microscopy. So when we talk about automation, it means that we have to have information that is both human and machine readable. When I talk about open science, it means it's open at time of publication. So we're not asking people to have everything available before publication. In terms of reproducibility, I think it's really important to consider including instrument quality control metadata information in our workflows. We need to have information that is findable by queries and searches, accessible by authentication, authorization, interoperable. We need to be able to connect microscopy images with genomic data, for example, and we need to be able to reuse our data with new hypotheses or new analysis tools. To achieve all of that, it is absolutely crucial that we embrace research data management, or RDM. This means that we need to capture data and metadata, and metadata is information about data. So organization, storage, preservation, and sharing are all crucial to achieving our goals. All right. So this can be done before. Voila. All right, so research data management before publication for everyday organization and working efficiently with our team and not spending time looking for the Excel spreadsheet on one computer and the data set on another computer, and post-publication for sharing and preservation. I strongly recommend that you have a look at the RMB model for research data management. RMB stands for Recommended Metadata Recommended Metadata for Bioimaging. And essentially, it's about classifying your metadata in terms of experimental design and sample preparation, microscopy, imaging, data acquisition, quality control measurements, data provenance, and so forth, and then image processing and analysis. In Canada, we now have research data management policies that are in place, and this means that scientists that are funded by the main 3 funding agencies in Canada have the obligation at time of publication to deposit into a digital repository all the information that support the conclusion of their publications. And that means data, metadata, and code. Now, luckily, the Canadian government did organize a national infrastructure to assist this. And this is called the Digital Research Alliance of Canada, or DRAC for short. And the mission of this organization is to provide support for all the research activity. So we have storage capacity for research data management. We have advanced research computing clusters, and they support software development. Now, Canada is a large country. We have data centers that are dispersed across the country. And these data centers are connected with high-speed connections, and this infrastructure is available to Canadian scientists. Now, in terms of data analysis, the way this works is that you're going to have your data in a storage component. And when you develop a workflow for image processing and analysis, you're going to connect to the cloud and submit your work to a job scheduler. And now this scheduler, depending on the type of computing infrastructure you need for your work and the size of your data set, is going to dispatch these computing jobs to various nodes and send back the output of your workflow back into storage. In the province of Canada, in the province of Quebec, where Montreal is located, we have 2 advanced research computing clusters, and one of them is at McGill University. And this is under operated by the provincial organization, which is called Calcul Québec or Compute Québec. So again, we have 2 computing clusters in the province of Quebec. And Calcul Québec offers a number of training courses for people interested in using the infrastructure of the Digital Research Alliance. So things like how to learn Unix, the Unix shell, command line operation, revision control with Git, Jupyter Lab, like Paul just mentioned earlier, GPU programming with Python, how to get started with the infrastructure, how to use Python for data analysis and visualization. More specifically for microscopy, there is a great open source software which is called Homero. Homero was developed by an organization called Open Microscopy Environment, the red circle in the middle. And this is really a fantastic tool for handling all the aspect of your life cycle for imaging. So you can store, manage, tag, query your microscopy data set. You can use Homero to visualize your microscopy, your multi-dimensional microscopy images, which is something very peculiar to microscopy that people in genomics and flow cytometry don't really have to handle with. And you can also perform quantitative analysis from within Homero. Now, OME has a lot of detailed instructions on how to install an Homero instance in your working place. And combining this information along with the information that the Digital Research Alliance of Canada has provided, a member of Canada Bioimaging, Steve Ogg, on the left side of the panel, Steve installed an Homero instance on the Digital Research Alliance cloud for Canada. So as a result, we have scientists, Canadian scientists now have access to a cloud-based Homero instance. I want to point out that Steve is not a computing scientist, he's a life scientist. And using these resources, he was able to get this cloud-based Homero instance. Now, we were also lucky because Claire Brown and Thomas Stroh in Montreal received funding from the Montreal Neuro Open Science Institute to further support the implementation of open, reproducible, and fair science. And we were able to hire people to assist scientists to upload data and metadata into the CBI Homero instance. And we automated the process for data upload at the same time as the metadata annotation process. And in the long term, we want to implement cloud-based advanced research computing via Homero. So if you know a little bit about imaging metadata, it's intense and intimidating and complex. We have 3 levels of metadata to deal with. One is sample preparation, microscopy equipment, and data analysis. So we wanted the challenge we had with our group was to not only capture all this information, but we also wanted to automate the process of capturing this information, and we were really, really challenged with that. Now, it turns out that we have a colleague, the group of Katerina in Boston, along with her colleague Alex. They had worked out the solution for doing exactly that. So working with them, we created a batch import in which we ended up having raw images, sample preparation metadata, and microscopy data right into the Homero instance. And come and get cookies, maple flavored cookies at the poster session, and I'll give you more details on the subject. So we're still not compliant for the Canada policy requirements because we still have to figure out how to link analysis data and code into this. It's not automated, but there's a way of doing it manually. Where you can attach your workflow, you can run script from Homero, and you can actually use Jupyter Notebook running on the Digital Research Alliance of Canada and do all of this in the cloud and being and hence become compliant with the agency. So I'm going to skip this slide, but Paul had a lot of point about virtual environments and Jupyters, and there are ways to get started on that. So do consider it very seriously. It's a wonderful thing to have if you want to go into big data, large data sets, and report or your imaging process. So we had, in conclusions, we wanted to foster open, reproducible, and fair science. And this was challenging, and it's complicated, it's time-consuming, and we managed to do some of it. But in the process, we came to realize that as a community, if we group the people, pair the people that have challenges with the people that have solutions, we can move further along. And as a result, a new user group was created with BINA. It's called Automated Image Management and Meta Annotation, or AIMM user group. And the idea here is to bring people at all scales, all level of the process, very beginners, or some people with more specific challenge, and get to have those people talk together in a non-intimidated and non-complex way, so that we move on and get organized and evolve despite the lack of funding around the world for doing all these things. So I'm going to end here and say a big thank to my teammates and all the funders that made it all possible. Thank you.
So thank you very much, Judith. I'd now like to invite our three speakers to come up to the front, and we'll have time for a Q&A panel discussion. Okay, so I think we're open for discussion. So Vlad, go ahead.
Thank you for the fantastic talks. The question, in many times then when there is a funnel, I see that biologists identify the problem and come to image analysts. How do you fill that gap? Sometimes they don't know where to go. Sometimes they don't know they have this capacity.
I think that you always know that you have a problem because you need to get a result. So if you need to get a result and you are not able to get it, I think that you know that you have the problem. So that's when I think that we can work to try to solve this.
Yeah, and I really think that this is where the role of the imaging core, in my opinion, comes in, because again, these are people that by the time the images are made, the scientists trust them, as well they should, because these are experts helping them out. And so I really think that imaging core folks can hopefully be able to bring more analysis into their cores, but even if not, can be consultants to tell, you know, the end user, like, hey, you might need to change this for your analysis, or your analysis would be fine, you just need to switch tools.
I think that ImageSC has been a fantastic tool, and I strongly recommend. But all BINA, Quarep, and it would be fantastic to count on the LABI and Latin America. I think the meetings we're having right now are spreading and synergizing and metastasizing. Sorry for the bad word, but networking, I think, is also very crucial.
So I have a question for Paul, or comment. I was super excited by all of your research online in Spanish, and it struck me there's like a way for BINA to work here in a two-directional way, right? You say you need to expand your network of experts in AI. So of course, people in the US could help you with that, right? They could help with online teaching and online support. At the same time, though, I mean, I live in New York, and we are, you know, every day, thousands of migrants coming in, all of whom don't speak English, but they mostly speak Spanish. And so I wonder how much you can help all the people in the US for whom English is a second language, and they would love to see these tutorials in Spanish. So have you reached out at all? Have you advertised to the US that you have all of this stuff online?
I think that I need to do that more. I think that I need to, with BINA, probably it's a really, really good connection. I have all these tools available, but I think that I have not advertised enough these tools, but I think that I have to do that. And I know that my network has to grow. I know that a lot of people in USA, for example, Beth, that she's doing an amazing job. So I have also from Latin America. I just talked with Federico. Yesterday we talked, and I think that we can make these connections. And I think that these meetings are really important to make all these connections that I think that are useful for me, are useful for the community, and are useful for everyone.
Really exciting. Thank you.
Hi, great presentations, everyone. So I'm going to ask a question to make things a little bit more complicated. I would love for all of our analysis pipelines to be integrated into something like Omero. It's a beautiful piece of software. But my analysis is not a script. My analysis is a very complex workflow where maybe I've gone into Cell Profiler, maybe I've done some sort of nightmarish Jupyter lab where I pulled in a neural net and done some complicated segmentation like Paul's describing. How do you anticipate Omero interfacing and interacting with all of these increasingly complex AI models, multiple pieces of software playing together? How can you bundle that up and make it actually reproducible?
I know that I've talked a lot with the OME team and talking about containerization and running containerized workflows, and I think that's going to be a big part of it going forward. I think that's definitely in their plan and in their roadmap, but yeah, having it then into something like Nextflow or SnakeMake where you can then have actually a whole workflow system is going to be critical. I'm also hoping that Cell Profiler, which the PR for the new Omero integration came in this week, because it can now talk to Docker containers and things as well, could be one of those bridges where you can make a Cell Profiler plugin for an arbitrary container. The whole thing comes through as a Cell Profiler pipeline too. Oh, can I explain what a container is? Yes, so containers is basically, think of it as an operating system in a box with some software pre-installed, possibly even data in there or metadata if you want it. And the nice thing is you install it once and only once, and then it works on Windows, Mac, Linux, essentially forever. There's a little bit that's technically wrong in that explanation, but it's close enough.
That's a great point, David. I want to point out that we're not necessarily focusing on Omero. Omero seems to be a good entry point, but yes, there are people that have developed and evolved without Omero, and we want to collaborate with those people so that we're not just focusing on Omero.
So we have a question from our online group.
Yes, just one quick comment, I think, about Paul's tools. We'd love to see them in the microscopy DB. Kate Phelps, who helps us run that group, we want to see them there for sure. Any language and materials is welcome. And then Elka asks, do you find that people from other Spanish-speaking countries attend your virtual sessions, or do you advertise them outside of Mexico? I think that's partially been addressed by Allison's comment. So do you find that people from other Spanish-speaking countries attend your virtual sessions, and do you advertise them outside of Mexico? So that might have already been answered, but if you could just reiterate.
Well, I think that I have not advertised enough. I think that I need to do that. But these workshops, for example, we gave one online workshop for people from Costa Rica. So I think that it's available from anyone in Latin America that the native language is Spanish. So I think that I have the video recordings. I think that we record all these workshops, and I need to upload these videos to BINA. I have to do that.
Just before the next question, I would make just a little plug for the microscopy DB, because I don't think that's come up in our conversations directly. I've seen it on a few slides, but it's a place where you can deposit your events, your training resources, your job ads, and then it feeds out to I think about a dozen now, different groups, the RMS, Canada Bioimaging, Africa Bioimaging. So you do one entry and then it populates in all those places. So highly recommended. And you can ask Nikki if you want more information at the coffee break.
Yeah, hi. Thank you for all the talks. Really great. I have like a more general question. Beth, I totally agree with you that we are always like separating image acquisition or microscopy and image analysis, and I feel that it should be always like more linked, and we should like integrate this in all our meetings or all our congress. The problem that I face, and I don't know if I am the only one, but in my institution, we don't have any, let's say, bioinformatician dedicated for image analysis. I feel that they cost a lot. So I think maybe it's like, because we are like linked to a university, we cannot really afford like having like multiple bioinformatician. So I don't know how we can like address this problem because I feel that we don't have enough resources and staff to do the job.
No, thank you for bringing that up. That's a really good point. So I think it has to be a multi-part solution, one of which is there are outside groups that you can sort of contract image analysis to. Our group, for example, we collaborate with Folks Anywhere. I know of at least a handful of people who are sort of independent image analysts that you can contract with. We've, as part of COBA, have been trying to get some, core director image analysis train the trainers workshops going for years. The major problem being like core facility people are so busy, they can't even take, a day or two to come do that. And so, I don't really know the solution for that other than, we've been trying to put, again, we've been grateful to the BINA EOEs. We've been doing cell profiler workshops as part of the EOEs, usually on either end. So we're trying to grab people when they're already out of the office to help do it. But it's hard for people who already have such limited time and so many demands on it for them to try to learn something else. But yeah, I think staffing for a full-time image analyst until we do a better job to figure out how to cost recover image analysis, which is a whole other thing, is really tricky.
Yes, maybe another quick comment is that do you think, because we also have like a pathology facility and a flow cytometry facility, and they all generate a lot of data, do you think it's I think I'm not the only one having also this type of facilities, and do you think one guy can deal with all this type of data, or it should be really separated? Or maybe it's like a command that we can have like
I think the devil's going to be very much in the details in terms of how much data, how much other stuff is that person trying to do in addition. But I think it's certainly possible in a facility that's, well organized and set up well, and this person like has dedicated time for that. But there's going to be times when that's true and times when that's super not true.
I'm just going to quickly add, again, if you want to go on public forum, that's one way of doing it. And you could also, as Beth said, hire people. So as you know, Cellavie does it, but there are other people doing it. There's Caroline, there's I know a guy named Brian, and maybe someone's going to be brave and start another independent consulting company. And maybe that's in the meantime, maybe that's the way to go.
Just to comment that imaging is not just microscopy, right? So they have preclinical and clinical imaging facilities as part of the larger group. And one of the great things is that they've already solved that problem in the clinical area. So by partnering with your clinical colleagues, if you're near a medical school, for example, often they have people because image data is data. And curating it has similar problems and solutions. So that's an option.
So Victoria Rapato comments, I've seen PhD PhD students that reach to their confocal microscope and their sample to get an image, but they have no idea how they're going to analyze the image after they get it, which means they have no idea what properties they want for their image. Has it happened to you that you have to say, the biologists, your images are not proper for analysis, go and get new ones? So I'm seeing a lot of nodding, but I don't know if the panel still wants to comment further.
Yeah, I think and that's part of why we hire biologists, because we want to not just have to say, these aren't good, go back. We want to say, these aren't good, go back and change X, Y, Z, and be able to sort of direct people to what's wrong and how to fix it. But yeah, garbage in, garbage out is unfortunately a big problem. And we always advise people to come to us early and come to us with the pilot data and things like that. It's, again, the cycle.
I would say if you take the habit of using research data management and start at point 0 on top and consider your experimental design and try to go through your whole cycle before actually getting on the microscope and preparing the sample, you're going to save a lot of time down the road.
So I think that is exactly the same for me. So it is go and back, and you say, okay, these images are not good for me. If you modify this, I can do something much better, and you are going to have a better analysis. And they say, oh, I only need to move this button, and you are going to get the image that you need. I say, okay, that's the solution for me, and it works really, really nice. So I think that this communication, we have to have communication as IE experts and biologists. So communication is everything.
I just want to add, I think for those of us working in imaging facilities where we often get to the researchers when they're at the start of the project, to really advocate for planning that whole life cycle of what do you want to measure and how are you going to manage your data. And then the pilot project, I think, is something we really need to instill in the community, that just do everything with a small set and don't do all of your experiments and then go for analysis. So.
It's for me? Okay. It's just a follow-up of a question that was online about education in Spanish for Latin American people. Initiatives like this start to coming out, and this year we released the NAPARI Latin American Bioimaging School, so people can check it. And the idea of having these schools is pairing with people from NAPARI to train new imaging scientists, and in the way in which we do, we combine aspects of language. So the written content is in Spanish, but we have experts in image analysis that can speak either Spanish or Portuguese that are already there supporting the professors for questions, for giving follow-up to the problems that the participants have. So I recommend to have a look in this kind of initiatives and in the next call just to jump in, because then we'll have a more connection in your own language.
Hello, hi, this is Katerina from UMass Medical School. So, I mean, I have just a comment, I think about, the issue with not having enough people in individual facilities to be able to do all of this stuff, even though we all want to do it. That's a very, very common issue. And the training can only get us so far. And sometimes, hiring people from the outside is a good idea, but if you don't have the money, you cannot do that either. So I think we need to, there is an advocacy effort going on right now to try to, we submitted a response for, a response to a request for information from the NIH, in which we're really trying to present the challenges to the funders of needing to invest more money in infrastructure, considering infrastructure also as, for example, distributed software, so image analysis, image data management, support hubs. Because if a person like, for example, Christina Baer at our institute who is running her own core would be able to plug in to a hub where she can get some software customization, a lot of different things, then it would be much easier for her to do it because she wouldn't be alone. She would be plugged in to a whole system of distributed software help and customization and automation. So I think there is definitely some stuff that can be done with the current resources, but we also have, as a community, collectively advocate for more funding and different ways of funding things.
I just wanted to circle back to something that Adan was saying earlier about having the resources in different languages. BINA's training and education working groups, as well as in image informatics, we've been piloting for the last about 6 months now an online system for translating anything that's marked down in Jupyter notebooks, anything that is sort of a structured text or YouTube captions. And it goes through automatic translation first, and then we just need language experts to fix where the automatic translation is wrong. We can do essentially unlimited content on that for at least the next few months, and then we'll reevaluate our license. If you have training materials, as long as they're open source license and you want to contribute them and you want us to help you translate them, come talk to me. If you want to be a translator and sort of check that translations are right so that a material can be in your language, come talk to me or come talk to Vanessa and Nikki, who also have access to the system. We really want to try and get the most out of it, and we can have up to 50 people at a time who are in the system as translators. And so we can absolutely add more content. We can absolutely add more people. We would love to sort of get the most out of this pilot system that we're doing and have it be valuable. And so far, it seems like it is going to be. So if you have material you want to be that is in Spanish and you want it to also be in Portuguese, come talk to me.
So just thinking back to Vlad's question about how do we bridge the gap between people that need or can make use of these resources and actually getting them to the resources. And I think it's something that we can maybe manage better, because if we're working on these models, normally there's a pioneer. I remember in the case of Paul, we had a group from the National Hospital who wanted help with, multiplex immunoanalysis, and we were working with one of the students, Sarai, and at the end of it, we said, well, yeah, okay, we got some great results, we published them. How much time did we save here? She said, oh, maybe 2 years, about a year and a half. Those sort of numbers, I mean, when you hear those sort of numbers, it's really impactful, and there's nothing more persuasive than the case study. It's actually, you can talk about these tools in the abstract sense, or come and use these tools, but when there's real people getting real benefits, and they're not having to do 2 years of clicking a mouse to get these results. So I think we need to work with our users and to ask them because these results are now being presented in conferences, they're being published. Sarai is doing her PhD. Well, she's got it now, but and she was doing her PhD exam. They don't have to. Maybe the context isn't right to explain the whole pipeline and how they got there, but if they can just mention that, if they can just put a quick point along, look, we did this collaboration with this group and it saved me 2 years of work. It doesn't have to be a big, long explanation, but it can be very impactful. We should work with our users to make sure that they're transmitting the idea that, these tools are transformative for their save them a lot of pain. I think we can help ourselves here, for sure.
All right, we have another question from Elka in the virtual audience. What can the role of microscopy facility managers be in research data management if we don't own the data?
Certainly, and again, I think Judith is going to be able to add a lot more to this. pointing people at best practices, pointing people to, again, like, so this is why Katerina does these amazing RDM EOEs, where she explains, here's how things can be set up, things like setting up a facility-wide or nationwide Amero instance so that somebody has a tool they can use. But
yeah, I really strongly believe that as a core facility manager, if you can sensitize your user about the importance of the image life cycle, that's already a huge step. And you can get started at different level, and you can go really, really deep, but you can make it simple to start with, and that's already a big improvement.
I just wanted a question for the panel in general. Are there things that the bioimaging networks like LABI and BINA could do that they're not doing already that would really move this along? Or if there are things they are doing that you haven't mentioned yet, maybe just to highlight those. Any thoughts? So are there things that LABI and BINA could be doing to help improve the communication and image analysis, research data management?
Well, I think that these meetings are really important. So these meetings here, LABI, BINA, are doing these meetings. So we are having this more regularly, these meetings. So these are really, really important. We can talk personally. So I had made, as I said before, many connections, and this is the most important. I think that they are doing a great job, and just continue with these meetings. So hopefully they are going to have some financial aid. So that's important because this is not cheap. So, but this is really important, these meetings.
the only thing I would say, and I think probably I'm preaching to the choir here, most of the people in this room are doing it, make sure your researchers know about, not just that you know about Microscopy DB, but that the people in your facilities that you link to things like Microscopy DB, to things like the image.sc forum, make it easy for your users to stumble upon the rest of us. And again, I think a lot of you already do. But let's make sure, like, these meetings are amazing and so many connections get made here and so many new ideas get made here, and let's make sure it leaves this room and sort of propagates. But I think, again, this is the choir here.
So I have a follow-up question for Beth. So regarding what you're saying about translators can offer to translate things for you and vice versa. So is this just applying at the moment to Spanish and Portuguese? This is also for Claire. This is like a question for the Global Bioimaging Initiative, really. Like, are you interested in also getting things translated into other languages? Like my image analysis guy is Indian, so I'm sure he could do that, right?
Yeah, so for the bioimaging guide, which again was the BINA Image Informatics and Training Education Group, is right now working to translate it into French, Spanish, Portuguese, Czech, German, Finnish, and Polish. And we haven't had any sort of non-roaming character other than Finnish is sort of non-roaming character. So, but we're happy to have any language where somebody wants to commit their time to say, I will review this, initial translated content and make a version and make sure that it's good in my language. At this point, we have the system down so that adding a new language to a material takes 10 minutes. So, and then the translator's time. So we want to make sure we respect that, but we've tried to do that by making it as easy as possible.
Great, thank you.
So I would just add, I think there's probably people in Canada Bioimaging who would be happy to work with you as the French proofreaders. So yeah, oh, they already are, Vanessa tells me. So we'll make this the last question, really. We need to wrap up. So.
Yeah, I really love the idea that, I really love the idea, like, as a core facility manager, we have to sensitize more our users to all the image life cycle. I am wondering if it will be possible to create through the BINA a printable poster with, like, we were talking a little bit with Claire yesterday, but you need help for your image analysis, and then you have like a QR code and they can access whatever resources. You need help for your whatever metadata, or I think it can be really helpful, and then we can have that on our core facilities.
It's a great idea.
I have another idea that extend that, but we know that Microscopy DB is a great resource, and maybe we could present. Could we present Microscopy DB, the resource, as like matching the REMBI models? Like, this is resource for metadata annotation for experimental resource. This is resource for analysis. This is resource for microscope. So maybe we could visually map the debate, the Microscopy DB with the REMBI cycle.
Excellent. I think that's a great spot to wrap up. We see how the community can engage and continue to help solve some of these challenges. So thank you for the engaging conversation and questions in the audience, and thank you for our panel.